more important. By the way, human and this one, plant reproduction, both are equally important. But human reproduction is smaller than reproduction in a plant, and the questions are being asked frequently. So I'll start with the human reproduction part, right? And again, the same way, we'll keep on teaching. In case if you have doubt, you have to ask me. So first and foremost thing, sometime the question I've been seeing from gestation period, and there are you need not to cram every gestation period, but there are few organisms whose gestation period you need to remember, right? And it is written on the screen. Gestation period of the rat. So the gestation period is that time period for which mammal carry baby, right? So the gestation period for the rat, that is the 20 days, and then the gestation period for rabbit, that is for the 30 days, human, that is for the 280 days in terms of days, and whale, this is 365 days, means for a year. Rhinoceros, it's more than a year, 5-10 days, and the maximum for the elephant, that is a 641 days, means 18 months, right? So these are the things which you need to remember, actually, right? You need to keep in your mind, because the gestation period of the rhinoceros have been asked, rat have been asked, right? Okay. Now the next thing is that sex organs, right? So there are three kind of the sex organ: primary sex organ, secondary sex organ, and accessory sex organ, right? What are the primary sex organs? So basically, primary sex organs are those parts from where the formation or production of gamete takes place. They are called gonads. So in male and female, respectively, the male have testis as a gonad, and female have ovary as a gonad. Male produces in testis, they produces a sperm, that is called male gamete, and the ovary is female gamete. Now, what are the secondary sex organs? The peculiarity with the secondary sex organ is that they do not produce any kind of the gamete or hormone. They do not produce gamete or hormone. Then what is their function? Their function is just they help in the transportation of the gamete, right? So out of them, there are reproductive genital tract and reproductive genital glands, right? They produces. So if they are not producing, what kind of the gland they are? I'll tell you what kind of the gland they are. Basically, they do not produce any hormone or any enzyme. So rather than they provide nutritional medium and the fluid for the transportation, right? And make the medium amicable for the this one for sperm, right? Okay. Now, what are the accessory sex organ? Accessory sex organ, they distinguish the male and female partner on the basis of physical characteristics, right? So the characteristic on which you can discriminate a male and female. Let's say in human you can differentiate that the humans male have beard, mustache, and the hair on the face, but the female generally they do not have, right? So that is a discrimination, and the body shape as well, right? In the same way, in any elephant you can decide on the basis of look, you can say this is female elephant or it's a male elephant. Female elephant bulky, heavy, more stronger, more powerful, and the male that is slim, trim, and it's looking better than female, right? So that's how you can discriminate on the basis of external look or morphological looks. So what are the accessory sex organs? The first thing is body build. Second thing is larynx. So you can see in male human Adam apple is visible, right? But Adam apple is not found in the female. And on the basis of their sound, you can discriminate male and female. Third thing, subcutaneous fat. Male have rough skin, while the female have a smooth skin. And the next thing, fourth thing, that is the hair, right? So hair on the body that we have, right? So primary sex organ, secondary sex organ, and accessory sex organ. I think it should be clear to you. Clear? Easy.
Yes, sir.
Very good. Taken the screenshot?
Yes, sir.
Very good. Now let's start with the male reproductive organ, right? You have to just understand and you have to remember a few things. I have a question for you before starting this one. Where does the storage of sperm take place? Tell me first. Where does the storage of sperm take place? That question have been asked in medical examination. Tell me. Abhinav, Alia, Hiram, tell me. Where does the storage of sperm take place?
Sir, in the epididymis.
Very good, excellent. So epididymis is the part where the storage of sperm takes place, but temporarily, not permanently, right? So that's good. That question have already been asked, and you will get this kind of a question. Just the asking question, it means that I want to tell you from where the question are coming, what kind of the question are coming from this topic, right? So look, the formation of sperm take place in testes. This part is testis, right? This part is called testis. The origination of testis takes place in from abdominal cavity. Abdominal cavity where visceral organ means this stomach and all that is placed, right? So formation of testis take place at visceral organ, right? At the seventh month of the fetus stage, or at the time of the pregnancy only when the person have not born, right? At the 7 month of the development, these testes come out from the abdominal cavity, right? So that testes come out. Why? Because for the formation of sperm, for the survival of sperm, the temperature of testis should be 2 to 2.5 degrees centigrade less than body temperature. Means it should be less than 37 degrees centigrade temperature. At 37 degrees centigrade temperature, which is body temperature, at this temperature, the sperm will die. So that is the function of a scrotum. This pouch in which this testis remains suspended, this is called scrotum. So scrotum I have written over here. Scrotum is called thermoregulator. Thermoregulator means it maintains the temperature, right? It's a thermoregulator. 2 to 2.5 degrees centigrade temperature lower than the body temperature. They maintain the body temperature 2 to 2.5 centigrade lower than body temperature, right? Now, the sperm production takes place inside the testis. These sperms are stored temporarily for very short time period in the epididymis. This part, this upper side part is called epididymis. So maturation of sperm takes place in epididymis, and the sperms are stored temporarily, right? I have written over here. Maturation of sperm takes place in epididymis, and it is stored temporarily here. These are the questions. Now, after that, these sperms are transported with the help of vas deferens. They reaches at this point. When they reaches at this point, here again there is a gland kind of the thing that is called seminal vesicle. Seminal vesicle do not produces any hormone and do not store the sperm. They produces seminal fluid. Seminal fluid provide the base for the sperm, right? Again, there is another gland that is called prostate gland. Prostate gland also produces the fluid which provide the nutrition and provide the development for the sperm, right? So both of the fluid they added and they provide. We'll discuss again this gland, right? And they provide the some base for this one, right? Now again, the sperm pass through with the same tube which is responsible from here. You can see at this point two pipe meet, right? The upper one, this is called urethra, right? Urethra is coming, and this urethra is this one. Ureter is coming, and from ureter, this is urethra. So urethra meet with the seminal vesicle, right? This is also a passage for the urine. Urine make urethra acidic. In acidic medium, if sperm will pass, they will die. So before the passage of sperm, first this bulbourethral gland or Cowper's gland. This small gland is called bulbourethral gland or Cowper's gland. It produces basic fluid and make the environment basic and neutralize the acidic effect. That's how the sperm are. That's how the sperm survive, right? So this is the function of this bulbourethral gland. So you have to remember the function of all of the gland and two things: this function of this scrotum and the function of this one, right? Where does the node and epididymis come? Is that clear?
Clear, sir.
Right. So you have to remember these things, which is written, or which I have written over here, right? Now, these gland, the function of these gland and all that, there are some more facts. Let's go much deeper. The same concept. First, the structure of testis that we have to remember. You don't need not to remember whole diagram. I'll tell you what you need to remember. Look. Now, this is the lateral section, LS, of the testis. In LS of the testis, you can see there are some covering around the testis. These covering are made up of some layer, right? The outer covering is called tunica vaginalis. You can see tunica vaginalis. Question comes from here. Tunica vaginalis. Tunica vaginalis have an outer layer called parietal layer, a cavity, and the inner layer which is called visceral layer. So parietal layer and visceral layer which is filled with the cavity. It makes the covering of this testis, right? Inside the testis, inside this tunica vaginalis, there is a tunica albuginea. This tunica albuginea is called capsule, right? Inside the capsule, there are these lobe kind of structure. These lobe kind of structure are called testicular lobe. Now, this is the question. How many testicular lobe can be found in a testis? The number of testicular lobe can be 200 to 300, 200 to 300 in a testis, right? That is the first one, right? Inside each lobe, inside each testicular lobe, there are some folded pipelines or these folded structure. These tubules, these folded tubules are called seminiferous tubules. The number of seminiferous tubules can be 1 to 4 inside a testicular lobe. Either it's 1 or maximum there can be 4 seminiferous tubules. Inside the seminiferous tubule, the formation of sperm takes place. The formation of sperm takes place inside the seminiferous tubule. From seminiferous tubule, these sperms are passed to this network-like structure. This is called rate testis. Rate means actually net. Rate means net, right? So this rate testis, they're forward here. After that, they, with the help of efferent duct, they passes through epididymis. We know that at epididymis, maturity takes place and temporary stage takes place, that we have already discussed, right? And then they pass through, right? So the number of testicular lobe, number of seminiferous tubule, that we need to remember. Now here this arrow shows that I have enlarged the diagram of testis. This is very important, very important, right? This diagram. You have to take the screenshot and you have to remember each and everything which is written in this diagram. It's very important. Look. Now you can see this structure. This structure is basically this, as you can see, right? This complete structure. This complete. You can see this complete structure. This is called basically, this is the transverse section of this one. It is the transverse section of a seminiferous tubule. In transverse section of seminiferous tubule, this is the outer epithelial membrane of the seminiferous, right? Inside this structure, you can see there are these kind of the globular cells, right? These globular cells are basically, they are called primary spermatocytes. So what is the primary spermatocytes, right? Or rather than primary spermatocyte, they are called primary spermatogonium. What are the spermatogonium? Spermatogonium are known as, they are made up of, what they are made up of. Spermatogonium are made up of germinal epithelium, right? They are made up of germinal epithelium. Germinal epithelium are a type of cuboidal epithelium. This is the question. So first and foremost, please note it down. Please note down. The germinal epithelium in every animal. Germinal epithelium. What are the germinal epithelium? The cells which produces In bracket you can write it down. Germinal epithelium, that is the cells which produces gamete. Germinal epithelium, that is the cells which produces gamete, are made up of cuboidal epithelium cell. Are made up of cuboidal epithelium cell in every animal, not only in human. So look, this spermatogonium, they are made up of germinal, they are called germinal epithelium, and they are made up of cuboidal epithelium, right? These germinal epithelium, spermatogonium, they convert into primary spermatocyte. Means they increase their DNA content, they increase their cytoplasm, become primary spermatocyte. These primary spermatocyte, then I have colored with the red color. They become a secondary spermatocyte. Division takes place, become secondary spermatocyte. The spermatocyte then convert into spermatid. Very important, spermatid do not have tail and head. You can see over here. Spermatid do not have tail and head, right? Then they further mature and become spermatozoa. You can see they are called spermatozoa. These one, spermatozoa. Let me write it down. These structures are called spermatozoa, or in general you can call them sperm, right? Spermatozoa or sperm are the metamorphosed cells which have the head, neck, and tail, right? Between these germinal epithelium, there are some cells, these rod-like cells, they are called Sertoli cells. Sertoli cells are also called nurse cells. Why? Because they provide nutrition for developing sperm, right? They provide nutrition for these developing sperm. When they convert, when they changes from spermatogonium to spermatozoa, the nutrition is provided by Sertoli cells. What is secreted by Sertoli cell? So first question, what is the function of Sertoli cell? Second question, what they produce? They produce ABP, androgen binding protein. First thing, they produces inhibin hormone. This question have been asked. They produces inhibin hormone. What is the function of inhibin hormone? They suppress the follicle stimulating hormone, right? That is the function of this one. Which one? Sertoli cells. Last but not the least, here between these pipelines, between these seminiferous tubules, right? Between the structure, this place where the seminiferous tubule, between this pipeline, let me color it as well. Just look here. I'm talking about this part. This part, the part which is between the this one, between the seminiferous tubule. So the part between the seminiferous tubules, this part, there are certain cells. The name of the cells are leading cells, right? They are called leading, right? So what is the function of leading cells? Leading cells have a specific function. They produces particular kind of the hormones, and these hormones are basically known as androgens. What are the androgens? Androgens are basically those hormones which provide a male characteristic to a male, right? Because of these hormone, a male becomes male. So androgen produces male sex hormone, right? For example, testosterone is a type of androgen. Okay, so remember the function of leading cell. Remember the function of Sertoli cells, germinal epithelium, right? That's it. Tell me, guys, is it clear or not? All of you. Abhinav, Alia,
Clear, sir.
Hiram,
Yes, sir.
clear? So please take the screenshot, and I expect that you will jot down in your notebook as well, right? Okay.
Yeah.
And you will get question from here. It's very important. Fine. Now, accessory sex organ. So as I told you that I'll again discuss the function of these glands which are present over there here. So again, the accessory sex organ. In the accessory, out of the accessory sex organ, there is seminal vesicle, right? Seminal vesicle. It's a gland-like structure. It do not store sperm, right? Sometimes it have been asked which is the true about the seminal vesicle, which is wrong about the seminal vesicle, right? So they do not store sperm. They produces a yellowish, slightly alkaline, means alkaline, slightly alkaline fluid for secretion. Seminal fluid contain fructose sugar. This question have been asked twice or thrice. Fructose sugar. Which sugar is found in semen? Basically, the fructose sugar is found in the semen, right? So seminal vesicle produces, and that sugar comes from the seminal vesicle. Seminal vesicle produces fructose sugar, right? Then they produces coagulating enzyme, right, which help the sperm the semen to coagulate, right? Ascorbic acid. This question have been asked. Ascorbic acid, right? Prostaglandin hormone, very important. From where the prostaglandin hormones are produced? Prostaglandin hormone are produced by the seminal vesicle, right? Hormone. The function of prostaglandin hormone, it it produces the contraction in the female reproductive tract. So what is the importance of this one? Contraction of female reproductive tract. What is the relevance of this one? Anyone of you? If prostaglandin increase the contraction in female reproductive tract, what will happen? And how it is useful? When the contraction in female reproductive tract, it increases the speed or motility of sperm inside female reproductive tract. Sperm itself swim, and along with swimming, contraction of female reproductive tract provide the motility to sperm, right? Ask question if you have any doubt, and keep on noting down, or you keep on taking the screenshot, whatever you want, whatever is suitable suits to you, but please keep on noting down. Prostate gland. Prostate gland, it produces thin, milky, slightly acidic fluid. It provides sperm nutrition, nourishment to spermatozoa. Corpus gland or bulbourethral gland. The question have been asked. Sometimes the answer will be bulbourethral, sometimes the corpus gland. Both are the same thing. So that you need to remember. The function of bulbourethral gland or prostate gland is they secrete alkaline mucus, right? They neutralizes the acidic environment of the urethra. Clear? Is that clear?
Yes, sir.
Very good. Oh, very good, Alia. You told me. Very good. Next, spermatogenesis. I have already told you that every heading is important here. You'll get the question, which of the following is diploid, which of the following is haploid, right? That kind of the question you will get. Again, testis. Inside testis, the seminiferous tubules, right? They are the seminiferous tubules. So let me write it down. Testis. They are seminiferous tubules. This is the transverse section of seminiferous tubules. Transverse section. In transverse section, you can see at upper side there are spermatogonia. These spermatogonia are made up of it produced from, or they are the modification of germinal epithelium or cuboidal epithelium. Spermatogonia can also be called sperm mother cell. Same thing. So which of the following is sperm mother cell? Is spermatogonia, and they are diploid. Then what happened after that? Mitosis takes place, means equational division. From spermatogonia, formation of spermatocyte takes place. The spermatocyte is also diploid. Again, the spermatocyte undergo meiosis, and secondary spermatocyte formation takes place. So spermatogonia is diploid, primary spermatocyte is diploid, secondary spermatocyte is haploid, right? Again, the meiosis takes place, second meiosis takes place, and formation of spermatid takes place. These spermatid metamorphose, right? They changes and provide formation of spermatozoa takes place. Spermatozoa is also known as sperm. There are Sertoli cells, and we have studied already the function of Sertoli cell. Sertoli cell provide the nutrition, right? Sertoli cell gives the nutrition. Okay. Clear? So they provide the nutrition. Now this is the spermatozoa. The upper part of a spermatozoa is called head, and rest part is called tail, and this is called neck. Like this part. This part is called neck. So head is made up of acrosome and nucleus, and they are a centriole. Neck is made up of middle piece. This is also called middle piece. This is made up of number of mitochondria, and this is tail part or flagellum. Right. Acrosome is filled with hydrolytic enzyme. Hydrolytic enzyme, they dissolve the wall of egg or previtelline membrane of the egg during the fusion time. That is the purpose of acrosome and these hydrolytic enzyme. Acrosome or hydrolytic enzyme is produced by the Golgi complex. That is the question that have been asked. From where does the hydrolytic enzyme or acrosome is produced? Acrosome or hydrolytic enzyme come from Golgi complex. Tell me, guys, is it clear or not? Go through with it and tell me. Is it clear?
Yes, sir.
Great, great. So what you need to remember, which of the following is function of Sertoli cell we already discussed, and here you just need to remember which is the haploid or which is the diploid. That you need to remember. One more thing, the life span of the sperm is only 24 hours. Why? Sperm remain alive for 24 hours. Nowadays, in the modern scientists, some of the scientists say that they remains 100% active for the next 2 or 3 hours only. But the lifespan of the sperm is 24 hours. Why? They do not survive after 24 hours. Look, the mitochondria is found at the neck piece part, this neck part. And these mitochondria produces the energy, right? These mitochondria, they produces energy. And this energy is utilized utilized for the swimming of this flagella, swimming of this sperm. Clear? Okay. Now come to the female reproductive structure. Female reproductive structure. This is the female reproductive structure. Female reproductive structure. Please remember over here whatever I am saying and what is the question. The question I've been asked from the wall of uterus, right? So this structure is called uterus, right? And the space inside uterus is called uterine cavity. Right? This space is called uterine cavity. Actually, this uterine cavity, when pregnancy takes place, uterine cavity become warm, and the expansion of uterine capacity can be 500 times. Means this uterine capacity can increase its size 500 times at the time of pregnancy. Means basically in a nulliparous woman, means what is the nulliparous woman? What is multiparous? What is nulliparous woman? Nulliparous, yet the lady which have not given birth to any child. Multiparous means the lady which have given birth to more than one child. Okay, so nulliparous woman, the space is only 10 centimeters square. At the time of pregnancy, this space become 5,000 centimeters square. Means the uterine cavity can expand, can stretch up to 500 times. Okay. The outer layer of uterus is made up of perimetrium. Is called perimetrium. Middle layer called myometrium. Inner layer called endometrium. Try to understand. Perimetrium is made up of epithelial tissue, right? Perimetrium is made up of epithelial tissue. You need to remember. Myometrium is made up of smooth muscles. What is the function of a smooth muscle? Any one of you, please tell me. What is the specialty of smooth muscles? Or what are the smooth muscles? Tell me.
Their function is contraction, right?
Right. What is the
And-
difference? Like you must have gone through that there are three kinds of the muscles: striated, smooth, and cardiac muscles, right? Remember?
Yeah. It's like smooth muscles are there in the digestive tract and the
Yeah, they are found there, right? So what is
and
their
the
behavior? What is their function? What it means? What is their behavior? They are type of. Can you control them? You cannot control them actually. They are involuntary, right? They are involuntary and they do not have, means they are automated control. They are not controlled muscles. You cannot control these muscles by with your own wish. Clear? They do not have any kind of the band. They are both of them tappers and they have single nucleus, right? So the most important thing that they are self-controlled. You cannot control these smooth muscles, means myometrium. Then there is one more layer that is called endometrium. Endometrium is the layer, the formation of layer with the layer which can be formed and can be removed, right? Every month, formation of new layer takes place. At the end of menstrual cycle, this layer breaks down and it goes out as the menstrual flow if pregnancy does not take place. Clear? So endo formation and okay, yes, sir. Formation and destruction of the this layer endometrium takes every month, right? It is made up of mucus, loose epithelial cell, and some blood capillaries. Is that clear? Fine. One more important. These ovary remains suspended with ligament, right? Ligament, they remain suspended with the uterus wall of the uterus and back side of the back, right? Means pelvic cavity, right? Means the pelvic structure. Okay. So this pipeline is called fallopian tube. Fallopian tube is further divided three parts: isthmus, ampulla, and infundibulum. Release or production of egg takes place in ovary. Ovary releases the egg. Ovary is not in direct contact with the fallopian tube. There is a distance. Then how does this egg reaches from the ovary to the fallopian tube? Actually, there are finger-like projection. They are called fembrae. This finger-like projection, they move like this way. And when they move like this way, they produce a suction pressure. Because of this suction pressure, this egg is sucked, right? When this membrane, they suck the egg, it reaches to infundibulum, then it reaches to ampulla, then it reaches to here isthmus. Between the ampulla and isthmus, here the fertilization takes place, right? So fertilization of egg takes place only in where? Ciliary muscle. Clear? Okay.
yes, sir.
Fallopian tube, again, one more important question. So first important question, where does fertilization take place? Fertilization, if you want to note it down, please, if you know, it's okay. If you don't know, please note it down. Fertilization takes place in fallopian tube between isthmus and ampulla. Fertilization takes place in fallopian tube between ampulla and isthmus. Again, ovary is a type of holocrine gland. What are the holocrine gland? Sorry, ovary is a type of heterocrine gland. What are the holocrine gland? Holocrine gland, let me remove it. Holocrine gland are the gland which produces only hormone. So ovary is a heterocrine gland. What are the heterocrine gland? Heterocrine gland are those which have other function other than production of hormone. So how the ovary is heterocrine gland? Remember, ovary is heterocrine gland. I repeat it again. Ovary is heterocrine gland. What are the heterocrine gland? Heterocrine gland are those which have other function other than producing hormones. How it is a heterocrine gland? Ovary produces egg. That is exocrine function. Endocrine function, it produces estrogen and progesterone, right? So function of ovary, production of estrogen and progesterone, two hormones. Other than production of hormone, it also have a function, that is the production of ova, right? That's why ovary is called heterocrine gland. Is that clear?
Yes, sir.
Okay. Look, one more important thing. Fallopian tube, the inner layer of fallopian tube, inner side, right? Inner layer of fallopian tube is made up of ciliary epithelium, right? Inner lining of fallopian tube is made up of ciliary epithelium. What is ciliary epithelium? They are this kind of the cells, epithelial cell which have hair-like structure, right? These hair-like structure. So they are called ciliary epithelium. These ciliary epithelium have cilia, right? It's called cilia, right? Ciliary epithelium have cilia, right? These cilia, they provide movement to the egg, right? They provide movement to egg. Provide movement to egg. So that is a function of ciliary epithelium, right? Again. So please take the screenshot of this one as well. Then I will tell you the structure of ovary. Again, this is the structure of ovary. So as I have told you that ovary remains suspended with the help of ligament. So ovary remains suspended with two kind of the ligament. You need to remember the name of that ligament. There are ovarian ligament. With the help of ovarian ligament, ovary remains suspended with the uterus. It remains attached with the uterus. And there are another ligament, suspensory ligament. With the help of suspensory ligament, it remains stuck with the pelvis. Clear? Is that
Yes,
clear?
sir.
Okay, fine. So first, formation of the primary follicles are always, all these follicle-like structure are formed, right? At the time of maturation. Inside the follicle, this follicle start growing, right? Under the influence of FSH, follicle stimulating hormone. So FSH hormone, it stimulate this follicle. This follicle are made up of cells. They are a granular cell, and inside there are egg, right? These are the growing follicle. In growing follicle, you can see, as you can see in growing follicle, in growing follicle, this is structure. This is called basically antrum. In growing follicle, some of the cell dissolve, and after dissolution, they make antrum. What is the antrum? Antrum is a liquid kind of the substance, right? This is a liquid kind of substance, this antrum, right? They produces a liquid kind of substance, and this is called antrum. Means some of the cell dissolve and they make the antrum. Formation of antrum takes place, right? This antrum is in the small quantity over here. It become in the larger quantity over here, right? This antrum develop, and the rest of the cell do not degenerate, right? You can see the rest of the cells, they do not degenerate. Okay.
So could you explain antrum once more?
Yeah, sure. Actually, what happened, you can see here in the growing follicle, this first growing follicle. In first growing follicle, there is an egg, and around them there are granular cells. These are basically granular cells, right? Round granular cells. Some of the granular cell at the upper part of the egg, you can see as egg moves toward the base, right? Lower part, and upper end, some of the cell, some granular cell dissolve and make a fluid-like structure, fluid-like substance. This fluid-like substance is called antrum. Clear?
Yes, sir. It's clear.
Actually, it helps in the rupturing of the ovary, right? So that is its basic function. Then what happened? This mature follicle, and the fully matured follicle is called Graafian follicle, right? This Graafian follicle is final and fully mature follicle, filled with antrum, some granular cell, and the egg. at the 14th day of menstrual cycle, this Graafian follicle rupture. And due to rupture of Graafian follicle, what happened? Egg come out. But after the rupturing, this follicle do not get destroyed. Egg is released, right? Now, this follicle do not destroy. The follicle regress, right? Regression of the follicle takes place. Means this follicle contract. Regression of follicle takes place. When this follicle regress, the formation of carpus luteum takes place. Now, please try to understand me. This Graafian follicle, where the egg is ejected from Graafian follicle, this remaining follicle is called carpus luteum. Now, this corpus luteum, what is the function of corpus luteum? Corpus luteum become secretory, means it start behaving like a gland. It is a cream color gland, right? It start behaving like a gland, and it produces some hormone. What hormone? They produces two kind of the hormone, progesterone and relaxing hormone. During the pregnancy, progesterone hormone is found, right? Progesterone hormone, please try to understand. Progesterone hormone maintain the pregnancy. If the progesterone hormone is stopped, then abortion will take place. That is called miscarriage. Generally, the miscarriage takes place after the pregnancy takes place because of the decrease in the level of progesterone. So that's why nowadays doctor keep on tracking the progesterone, and they give the injection of progesterone if it is producing the less amount inside the females, right? At the end of the pregnancy, after the 9 months, right? At the end of the 9 months, when the pregnancy is getting terminated, means parturition will take place, birth of child will take place. On that time, this corpus luteum will produce a relaxin hormone. Clear? Remember or not? Got it this point or not? Please try to understand. Got this point or not? I am stress. I am putting stress. At what point you miss it, right? And these are the questions. Very important. What happened? Normally, in female, unmarried female, where pregnancy do not take place. So, at the 14th day of egg is released, but egg is not fertilized, right? Then what happened? This corpus luteum get destroyed, and production of this progesterone stop. Due to stoppage of progesterone, this is the a question asked multiple times in NEET. Due to stoppage of progesterone, the endometrium do not remains maintained. To maintain the endometrium, progesterone is required. When corpus luteum degenerate, it stop working. Progesterone, release of the progesterone stops, and endometrium start getting destroyed, right? Endometrium start getting destroyed, right? And that's why the menstrual outflow comes. When pregnancy takes place, corpus luteum do not degenerate. It keep on producing progesterone. Endometrium remains permanent as a permanent wall, and it maintains the pregnancy. Did you got my point or not? If you want me, I can dictate. If you want to note it down, is it okay?
Yes, sir.
Iram, okay?
Yes, sir. It's okay.
Okay, great. So this was the function of this one. Then this is regressing corpus luteum. Then corpus luteum degenerate normally, and there is a membrane in the reproductive tract. This membrane is called hymen, and this is perforated membrane where menstrual outflow can come out, right? And there is a structure which is rich in the neurons. This is called clitoris, and clitoris is the rudimentary, right? It is the vestigial symbol of penis. It is the vestigial penis in female. Right? And that is the anatomy of external female genital organ, right? The outer level is called labia majora. Then inner layer is called labia minora, right? And this is urethral opening, from where the urethra open, right? And this is vaginal opening, right? And hymen layer is found here actually, and this is a clitoris, and I have told you, right? And there is a one more gland which is found above the clitoris. This is called bulbourethral gland or Bartholin gland, right? It is a vestibular gland, right? Bulbourethral gland. It produces some. It produces uh, uh, a substance, mucus-like substance, which maintain the environment of this vagina. So you have to remember the name of bulbourethral gland. It have been asked. Bulbourethral gland. Any question from female reproductive structure? No question.
No, sir.
Okay. Now, oogenesis. Spermatogenesis we have got. Now I have a question. Before explaining oogenesis, I have a question with you. What is the basic difference between spermatogenesis and oogenesis? Please tell me. Any intelligent person?
The start of oogenesis takes place in the fetal life, and the start of spermatogenesis takes place during puberty itself.
Very good. Excellent, Heather. You are late, Heather, today. What happened? You
But
are
actually,
too
there's
late.
some work going on at my home, so that's
Okay. Fine. No issue. Right. So very good. Excellent. Excellent. Actually, spermatogenesis start after the puberty, means after 11 year age of a male child. But oogenesis start at the fetal stage only, when a female baby inside the womb of its mother, right? Okay. As I have already told you that formation of any gamete takes place from cuboidal epithelium, right? So again, the formation of these egg takes place from where? The germinal epithelium. Germinal epithelium. And formation of all these oogonium takes place from germinal epithelium, right? And germinal epithelium are a type of epithelium. This is the name of epithelium. And what kind of epithelium? It is a cuboidal epithelium. It's a cuboidal epithelium, right? Germinal epithelium is a type of cuboidal epithelium, right? Just a minute. Fine. So, oogonium, this is germinal epithelium, it's cuboidal epithelium, and they produces the formation of oogonium takes place over here. When it is written over here, at female fetus stage, mean unborn female baby, right? Then what happened? These oogonium are, this is called again, one more thing, oogenesis have been divided in three phases: multiplication phase, growth phase, and maturation phase, right? Again, in multiplication phase, the multiplication phase takes place in the unborn female body only, means at the fetus stage only. Here the mitosis takes place. So egg mother cell, oogonium, and the formation of oogonium or egg mother cell taken place from germinal epithelium, they divide through the mitosis, and formation of additional oogonia takes place. Then what happened? From this point of time to achievement of puberty, what happened? The growth phase takes place, right? It's a very long phase, right? So at the growth phase, what happened? Primary oocyte is produced. Primary oocyte is also diploid cells. So what is the difference between oogonium and primary oocyte? Actually, the primary oocyte have almost double of size of oogonium. It have double content of genetic content, means DNA, and double protoplasm, right? And what happened? It occurs each month of the ovary female after puberty, right? After puberty, at every month, one egg get mature, right? Inside the ovary, there are certain cluster of oogonium. Those cluster of oogonium are known as egg nest. Out of this cluster, one cell, it increases cytoplasm, it increases its nuclear size, right? And it becomes the primary oocyte, right? Primary oocyte undergoes the meiosis, right? Meiosis takes place, means first resting phase takes place, right? In meiosis, first unequilateral division takes place. Normally, when a normal meiosis takes place, as we have seen in the case of this spermatogenesis, formation of two sperm cell take place. But here it's not like that, right? Here, because there the cells are of both equal in size. Here the cells of different size. One small cell is formed, that is called first polar body, and another larger cell is formed, that is called secondary oocyte. Secondary oocyte is released. So at the 14th day of menstrual cycle, try to understand. At the 14th day of menstrual cycle, the egg which is released, we cannot call that mature egg. That is a secondary oocyte. That is not actually egg, secondary oocyte, right? At the time of ovulation, that is the question that you need to remember, right? What happened when the meiosis second takes place? When the egg fuses with sperm, right? If the egg fuses with sperm, then it will undergo secondary meiosis. Else, it will not undergo secondary meiosis. Secondary meiosis, that is a question have been asked many times. I have seen many times this question, that when the meiosis second takes place. When the meiosis second takes place, meiosis second will take place when the fusion of sperm takes place. When the sperm will fuse, what will happen? The first polar body divide into two small cells. Again, unequilateral division will take place. Secondary oocyte will divide into two cells, a polar body and a mature ovum. Look, at the mature ovum, what happened? Plasmogamy taken place. Means protoplasm of sperm and egg have fused, but nucleus yet not fused, right? At the mature egg, the meiosis second taken place, plasmogamy takes place. It means mature egg have two nucleus, n plus n, right? Again, after some time, the means after a very small time duration, the nucleus also fuses, means karyogamy takes place, and formation of a zygote takes place, right? This is the all about the formation of egg or oogenesis. Please ask me if you have any doubts, and please take this screenshot as well. Is that clear? Okay. Come to the menstrual cycle. It is also important. What you need to remember, I'll tell you. Look, when the menstrual cycle goes on, changes take place at two places, right? During the menstrual cycle, the physiological changes take place at two places. First in uterus and second in ovary, right? Both of the things have been shown over here. Look. So the menstrual cycle, this is ovarian cycle. This ovarian cycle, this is number one. It is, these diagram shows the change in the ovary which is taking place. So we have already understood that and we have already studied that. This is uterine cycle, means these are the changes which are taking place in endometrium. Okay, guys, look. This is the body temperature. It have nothing to do with that. You have to remember these things. Look, from anterior pituitary, from where hormones are released. From anterior pituitary, hormones are released. The first hormone is called follicle stimulating hormone, FSH. This is FSH. So you can see the peak of FSH, means maximum amount of the FSH is released at the 14 days. After that, the FSH level drops, right? What is the function of FSH? Its function is growth of follicle. Primary follicle grows into growing follicle into Graafian follicle, right? And at the 14th day, this LH, luteinizing hormone, also increases, right? when luteinizing hormone, so which hormone is pushing this luteinizing hormone to release? Estradiol, right? Estradiol stimulate the luteinizing hormone. Luteinizing hormone which reaches at the peak, you can see the ovulation take place ovary from this this follicle, right? Ovulation, so which hormone is responsible for ovulation or release of ovide 14-day? That is the LH, luteinizing hormone. LH and FSH both are released from anterior pituitary. First, FSH responsible for development of the follicle, LH responsible for release of the ovum, right? Again, now what happened? The LH also reduces, the level of FSH drops, and when LH and FSH are at the top point, means at maximum level, the body temperature of the female body will be maximum. Then LH and FSH drop, its level drop after ovulation because they have completed their work, their job. Then the temperature gradually downs, right? Again, ovarian hormone. Now, what is happening in the ovarian? From the ovary, a hormone is released. The name of two hormones is estradiol and progesterone. Estradiol have two functions. First, it pushes, first thing, the function of estradiol. Estradiol is stimulating the luteinizing hormone and inhibiting the FSH, follicle stimulating. It's suppressing the FSH, right? LH increase, FSH suppress, right? Then estradiol have these two functions. Then what is happening? Again, after the ovulation, you can see progesterone is released in larger amount. This progesterone is required for the maintenance of this layer, endometrium layer, right? This layer develop, and along with the endometrium layer, these mucus layer or some of the loose cells of epithelium develop, and along with that, blood capillaries also develop. These red color thread are blood capillaries. When fertilization do not take place, look, try to understand from where it is developing. It is from where it is coming. I have already told progesterone is produced by corpus luteum, right? You can see here, when fertilization do not take place, corpus luteum degenerate. When corpus luteum degenerate, the release of progesterone is stop. If the release of progesterone is stop, this membrane, this layer, endometrium, is start getting destroyed, because to maintain this membrane, progesterone level is required. But when fertilization do not take place, corpus luteum die and progesterone release stop. When progesterone release stop, this membrane start getting degraded, and when this membrane degrade, these blood capillary rupture. Because of the blood capillary rupturing, along with this, these loose cells, the blood also oozes in the menstrual outflow. So at the deficiency, this is the question have been asked multiple times. Due to the deficiency of progesterone, I have written over here. Due to the deficiency of progesterone, this membrane, endometrium getting destroyed. Endometrium get destroyed. Right? Tell me, guys, is it clear or not? Go through with it. Is it clear or not? If it's not clear, let me know.
It's clear, sir.
Very good. Fine. So you have to remember, you will get the question. As I have told you that this question, which kind of the question I've been asked, and there are some factual things. A female is fertile for the 32-year duration, means from the menarche to menopause. Every female releases 13 eggs in a year. FSS is it is released from the it is responsible for the maturation of primary ovarian follicle. And one more important, what is the function of SCG? What is the use of SCG? Can you tell me? What is the use of SCG?
It is released from the hypothalamus, and it acts on the ovary to release, on the anterior pituitary to release LH and FSH.
Okay. Is it clear? Sorry, is it? Are you confident with that?
Sir, it helps in the
Human
secretion
chorionic.
of pellets and
Read it out.
human chorionic gonadotropin.
It's not just gonadotropin. Look, you must have seen in the market there are some pregnancy strips, right? Females are using that to confirm the pregnancy. Those pregnancy strip recognizes they just get red, they changes the color due to presence of HCG. When a female is pregnant, then HCG is released, and this human chorionic gonadotropin, this HCG is released and it start coming out with the urine. And because by the testing the urine, right, the pregnancy can be confirmed. Actually, this HCG confirm the pregnancy. It is released from the placenta. Clear? HCG is the confirmation of pregnancy, right? So pregnancy strip, they recognizes the HCG, human chorionic gonadotropin. We'll study its role in like next 50. Everything clear to all of you?
Yes, sir.
Yes,
One
sir.
more important thing. Yes. Any question? No. One more important thing. What is the composition of those pregnancy pills, eye pills and all that? Any one of you?
Sir, composition of?
Pregnancy pills, eye pills and all that.
Sir, they're made up of a mixture of estrogen, progesterone.
Very good, very good, very good, very good. Good. Yes, you are absolutely right. Fine. Look.
Sir,
Here
is it
the
progesterone or progestin?
progesterone.
Progesterone. Okay.
And there are some different composition of progesterone, basically. Look, ovulation takes place at the 14 days. Now after the ovulation, ova is released. Now fertilization takes place. So when the fertilization takes place, we count it 0 day. You can see from the fertilization takes place, we count it 0 day. Then the karyokinesis takes place, new cytokinesis takes place, then karyokinesis takes place, and the day first, one cell stage takes place. In the day 2, this cell divide into 2 cells, right? And the day 3 and 4, they divide into 4 cell stages. Day 8, right? This is uncompacted morula stage. Morula terms means like mulberry, right? It is 8 cell stage. It is called morula stage, right? Then at the day 4, it start roll out from the fallopian tube, right? And day 5, day 4, day 5, day 6, day 7, day 8 or 9, implantation takes place. Implantation means it will stick with the endometrium. It will go deep inside the endometrium wall, right? And this stage is called blastocyst. Only you have to remember these two things. At the 4th day, it will roll out into uterus. At the 8th and 9th day, implantation takes place. And first cleavage takes place at the first day. These are 3 things which you need to remember. Clear?
Yes, sir.
This is basically the structure of egg when egg is released. When egg is released, it's not just clear kind of. There are some cells which remains stuck with this egg, and they are called the outer cell. They are called corona radiata, and then there is an inner membrane. This is called zona pellucida, right? And then there is a membrane called previtelline membrane, and then there is cytoplasmic nucleus, right? And when the sperm fuses, then sperm with the help of acrosome, right, the hydrolytic enzyme of the acrosome, it dissolve the zona pellucida, zona radiata, then zona pellucida, then pre-vitelline membrane. So that's how it penetrates inside that, right? So egg is basically released like this stage at this. Now sperm egg interaction. What is the polyspermy? Polyspermy. Polyspermy is fusion of multiple sperm with an egg, which is prevented in humans at least, right? Fusion of multiple sperm in egg. When one egg is when sperm fuses with an egg, then the other sperms are rejected. They cannot fuse with the egg. Only one sperm which will reach first, it will fuse with the egg. Look, this is the wall of egg. The inner wall of egg, there is the egg cytoplasm, and inside inner wall there are these these kind of these globular structure. They are called cortical granules, right? Outside there is a vitelline membrane, right? There is a egg plasma membrane, and on the egg plasma membrane there is a pre-vitelline or vitelline membrane, right? And at the pre-vitelline membrane, at the outer side, there are found some protein receptor which recognizes the sperm, and then the jelly coat, which is made up of zona pellucida, zona radiata, right? Now the sperm is fusing. When a sperm comes for the fusion, sperm head is made up of acrosome. Acrosome have hydrolytic enzyme. Acrosome releases hydrolytic enzyme. This zona radiata, zona pellucida, and all that, this is released. Right? This is dissolved. After dissolution, the this nucleus fuses. When the fusion of nucleus takes place, you can see that this cortical granule dissolve. Dissolution of cortical granule form a pre-vitelline. It gets filled in the pre-vitelline space, right? And they changes and make a cortical granule membrane, right? Make a membrane-like structure. They changes the polarity of membrane. Because of the change in the polarity of membrane, another sperm cannot fuse, right? So formation of cortical granule membrane block the polyspermy, means block the fusion of multiple sperm, right? So polyspermy is prevented by the cortical granule membrane. Polyspermy is prevented by cortical granule membrane formation and change in the polarity of membrane, right? So this is the fusion of sperm and egg. Tell me, is that clear to all of you?
Yes, sir.
Polyspermy, which prevent the polyspermy, this question have been asked. That's why I have written down the both of the option. Sometimes the option will be this cortical granule membrane. Sometimes the option will be like change in the polarity of membrane. Zygotic cleavage, fertilization taken place. How does the zygote cleavage, right? You have to remember just this structure, the thing which I could tell you. First cleavage, then 2 cell, 4 cell, then 8 cell stage. And we know that after 8 to 16 cell stage, this is called marula stage, the mulberry-like structure. After marula stage, it again cleavage will take place, 32 to 64 cells and more. The blastula formation will take place. What is the peculiarity of blastula? Inside there will be a hollow structure filled with the fluid. This is called blastocele, right? Blastocele or blastocyte cavity, right? The cells which are surrounding the blastula or the cells which make the surface of blastula, they are called trophoblast cell, or this membrane is called trophoderm. Again, what happened? Again, further development take place. This trophoderm, it start intruding inside, inside like this way, right? And This process is called gastrulation, right? This process is called gastrulation. It intruded inside, right? And due to intrusion, formation of two layer takes place. The outer layer, which is made up of trophoblast cells only, outer layer is called ectoderm. Inner layer is called endoderm. Between the ecto and endoderm, there is a blastocele, right? In future, this blastocele also become filled with a cell, and this is called mesoderm, right? And there is a pore-like structure. This is called blastopore, right? And this is the gastrula stage. With the help of gastrula, at the gastrula stage, it make the permanent connection with the this blastopore make the permanent connection with the uterus wall, right? And after that, the formation of placenta takes place over here, right? So this is ectoderm, mesoderm, and endoderm. That's how we are the triploblastic animal. Formation of whole body takes place from these all three layers: ectoderm, mesoderm, and endoderm. Tell me. Is that clear?
Yes, sir.
So could you explain it once more?
Yeah, sure. Fine. Once again, look, zygote formation takes place, then zygote cleavage, and formation of a mulberry-like structure takes place. Mulberry-like structure is called marula, right? It may be 16 to up to maximum 32 cell stage. Marula can be up to 32 cell stage. After 32 cell stage, we cannot use the term marula, right? After marula, so first stage is marula, second stage is blastula. Then again, further division of the cell takes place, and formation of this ball-like structure takes place, right? It is like a tennis ball, right? It is hollow from the inside, but tennis ball is hollow inside and it's filled with the gas. But in place of the gas, here in blastula stage, there will be some fluid, right? So this cavity, this space is called blastocele. Blastocele or blastocele cavity. This blastocyst cavity is filled with fluid, right? And it is surrounded by a layer. This layer is called trophoderm. Trophoderm is made up of cells. These are called trophoblast cells, right? Then what happened after this one? What happened? If gastrulation takes place, due to gastrulation, this way, this part start penetrating inside, right? It start going inside. When it start going inside, it start folding like this way. When it will fold like this way, this process is called gastrulation, right? After this process, formation of gastrula stage takes place. In gastrula stage, look, this yellow color structure is nothing but it is made up of due to the folding of this wall only, right? The yellow color structure, it is called endoderm. This layer, it make a layer called endoderm. The outer layer, this one, it is called ectoderm, right? And between ecto and endoderm, this blastocele cavity, it is still called blastocele, but it get regress. Further, this blastocele also get filled with the cells, and this blastocele make a cell layer that is called mesoderm. So the fully mature gastrula is made up of three layers: ectoderm, then mesoderm, and then endoderm. Is it clear?
Yes, sir.
Fine. Now this diagram is also very, very, very important. You will get number of the question from the placenta and the function of placenta, right? Okay. Further, this blastula get gastrula develop, and as I have told you that gastrula pore, it make the placenta. This structure is called placenta, right? And this placenta is meant for taking the nutrition from the uterus of mother, right? Now the cervix part that get closed, the cervical part that get closed with material that is called clog. And then what happened? The formation of a membrane-like structure takes place around it. The membrane-like structure is called amnion, right? Amnion. Inside the amnion, there is a fluid that is called amniotic fluid. Amniotic fluid is, you can say, it's a urine of this fetus, right? Amniotic fluid provide hydration to uterus, developing uterus. It provide the nutrition, oxygen, and other required material. And in place of the it takes back the excretory material, carbon dioxide, and all that, right? So transfusion takes place through this amniotic fluid, and it also prevents this fetus developing fever protect developing fetus from the jerk and all that, right? Then there from the umbilical cord of fetus, there is a placenta. This structure is called placenta. Placenta also has a yolk sac. Yolk sac is a rudimentary structure, and it tells that formation of mammals or formation of these viviparous animals taken place means evolution. We have evolved from the oviparous animals, right? Again, the placenta have some finger-like projections. These finger-like projections, they get they goes deep inside to the wall of the uterus, right? And these finger-like structure are called chorion, right? And this you can see this is called chorion, right? And these fingers are called chorionic villi, right? So chorion or chorionic villi, it take the nutrition, but transfusion of the blood do not take place between the fetus and mother. Sometimes accidentally transfusion of blood takes place. Due to that, the jaundice takes place in the developing fetus, right? If transfusion takes place, right? And this is the umbilical cord. This structure is called placenta, right? Fine. Now, what is the function of placenta? That is a question for your medical examination. Placenta secrete HCG, human chorionic gonadotropin. I have discussed few minutes back, right? Which is which is the test for the pregnancy, right? It releases estrogen, progesterone, human placental lactogen. These are the 4 hormones secreted from the placenta. Exchange of the gases, nutrition, and exchange and excretory material takes place through the placenta. Amniotic fluid, it prevents from the jerk, help in fetus movement, prevent from bacterial infection, and it maintains the temperature and pressure. That is a function of amniotic fluid. Parturition means the birth of the child, right? When birth of child takes place, which is responsible for birth of the child? I have told you that the middle wall, middle layer of the uterus is made up of what? Involuntary muscles. What kind of involuntary muscle? Smooth muscles, right? So endometrium, myometrium, endometrium, myometrium, right? So the myometrium is made up of these smooth muscles, right? At the end of pregnancy, at the completion of 9 months, at the termination of pregnancy, release of a hormone takes place, oxytocin. Oxytocin hormone is responsible for expelling the baby outside. What it do? It start contraction in the smooth muscles. Due to contraction of smooth muscles, baby is expelled outside. Means pressure is created and baby is expelled outside. This process is called parturition. So function of oxytocin, expelling the baby, right? At the time of birth. After the birth, it is also responsible for milk ejecting, means production of milk in the mammary gland. That's why oxytocin is called milk ejecting hormone. This question has been asked twice. Milk ejecting hormone. Which is the milk ejecting hormone? Oxytocin is milk ejecting hormone, right? It activates the mammary gland and production of milk take place in the female which have given birth to young one, right? Help in expelling the baby. Again, there is a hormone that is named prostaglandin hormone, right? It is released from the placenta. This prostaglandin provide more contraction to the uterus. Means prostaglandin and oxytocin, 2 hormones are responsible for expelling baby or the birth of baby or parturition. This is all about this process. Tell me, guys, is it clear everything, or if you have any doubt, please ask. But whatever written in this screen, you have to remember everything. Every point is the question, believe me. When you will do the previous year question, you will find that whatever the point I have told you, they are the question, they have already been asked. And there are probability, and there are some point where the probability of being asked as a question. Is it clear to everyone? Heather?
Yes, sir.
Abhinav? Iram? Alia? Hope you must have taken the screenshot as well. Breast, just these are the just rib structure. And after them, there are some alveoli-like structure. They are called mammary lobes, right? Where the production of milk takes place, right? These are the mammary lobes, right? And these are the fat globules. And mammary alveoli, where the production of milk takes place. And then this milk come out through the mammary duct. Mammary duct make more wider duct called ampulla, and from ampulla there is a lactiferous duct through which the milk come out, right? And one very important thing which have been asked as a question, mammary glands are just modified sweat glands. That you need to remember. It's very important point. And after birth, prolactin is released. Prolactin is responsible for production of milk, right? Oxytocin, it is responsible for release of the milk, right? So prolactin, it is responsible for production of milk. Oxytocin, release of the milk. And there is a after the birth, after the parturition, for next 3 to 4 days or 2 to 3 days, for yellow colored milk come out of the new mother, and that the name of that milk is colostrum. Colostrum is very important milk, and every doctor suggests that the colostrum must be provided or feed to the young one, because it is thin yellowish fluid colored fluid, and it's milk in fact, and it provides nutrition, right, to young one. And the more than nutrition, it provides the disease fighting capability to the young one, because the this milk, it contain the antibiotic capability, or the disease fighting capability, right? It is passive immune system. It make the passive immune system. This is all about this chapter. So we have completed this chapter, human reproduction, and after the class, revise this chapter at your home, and do the previous year question. As I say that attend the previous year question which came after 2018, all the questions which came after 2018. And if you have any doubt, note it down and discuss with me in next class, right? So this is the time to wind up class. I think there should be some another class, right? So bye-bye, take care, have a good day, and do come with a question. Go through with the NCRT, go through with the previous year question paper, and go through with the PPT which I discussed with you. And still, if you have doubt, write it down in your pen down it and ask me next one, right?
Sir, I
Yes, sir.
had two doubts from the molecular
Yeah.
basis genetics
Yes,
chapter.
yes. Tell me.
I couldn't understand this packaging of the DNA and the nucleosome part of that chapter.
Packaging of DNA and nucleosome part. So can we discuss in next class, because you know your time is over.
Sure, sir, sure, sir.
Actually, some faculty might be waiting. Otherwise, we have discussed already, and we'll discuss. So what you have to do, note it down in next class. In the before starting the class, ask me, right?
Okay, sir.
Fine.
And sir, we don't have any other class today, sir.
Don't have any other class?
No, sir.
Okay, so I'll tell you right now. No issue. I thought that there must be, because I have 5 minutes, so I can tell you right now. Packaging of DNA, molecular genetics. What is this? Molecular genetics, packaging of DNA. Yes. Look, at the time of when the cell division takes place, so condensation takes place, and the packaging of DNA is meant for only the division of the cells, or means the division of the chromosome, right? That's when that's how the that is the purpose behind packaging of DNA, right? So first, the width of this DNA is 20 angstrom. It will start compressing, right, like this way, and after compressing, this compressed DNA, it make a 2.5 round of a protein octamer, and this protein octamer is basically made up of histone protein. So 8 histone molecule make this structure, and there is a 2.5 round of the DNA, and this structure which is made up of protein octamer and 2.5 round of the DNA, it is called nucleosome, right? Okay.
Okay, sir.
When nucleosome, when number of nucleosome are found in a thread like this way, these are the nucleosome, and between the nucleosome, this DNA which connect a nucleosome to another nucleosome, this is called linker DNA, right? So this structure is called chromatosome. This question have been asked many times. What is chromatosome? One more thing, there is a similar word. What is the chromatophore? What are the chromatophore? Even this question have been asked. Chromatosome is a beaded structure made up of nucleosome. What are the chromatophore? Blue-green algae do not have chloroplast. In place of chloroplast, for the photosynthesis, they have precursor of chloroplast called chromatophore. Clear? Chromatophore is found in blue-green algae meant for the photosynthesis, and chromatosome is this structure. After what happened, these chromatosomes, they make this kind of this structure, which have the width of 30 nanometer. This is called solenoid, right? So next packing is solenoid. Further, what happened? This solenoid again make this kind of the structure, and this is called chromatid, right? This is called sister chromatid tag, right? This the width of this fiber is 300 nanometer, right? After that, it further condense and make a chromosome, and every arm of the chromosome, right? Every sister chromatid is 700 nanometer thick, and the thickness of chromosome is 1400 nanometer, right? This is the condensation of the DNA or packaging of DNA. So you have to remember nucleosome, chromatophore, solenoid, and chromosome. 4 structures. Is it clear, Abhinav?
Sir.
Okay. Any more question?
So the structure of nucleosome, can you explain it quickly?
Yeah,
once?
sure. We have discussed the structure of nucleosome as well. This one. The nucleosome is made up of histone octamer. So there are 4 kind of the histone: S2A, S2B, S4, and S3. One copy is at upside, one copy is at downside, right? And this is DNA, which make 2.5 circle, right? And then this DNA is negatively charged, histone is positively charged. Both are stuck with each other, but it can slip. So further providing its firmness, there is one more histone called H1 histone. H1 histone give it stability, right? And this structure is called nucleosome. The width of nucleosome is 6 nanometer, and the height of nucleosome is 6 nanometer, and width is 10 nanometer, right? And there are 5 kind of the stone: S2A, S2B, S3, and S4, and H1, right? Histone is positively charged. This question I have been asked, and it is still important for your NEET examination. The histone is positively charged because it is made up of lysine and arginine amino acid, rich in lysine and arginine amino acid. Clear?
Yes, sir.
That's it.
Yes, sir. Sir, and there was one experiment in the Hershey and Chase experiment.
Yeah.
In it, there was the downside of it was somewhat. It was written centrifugation and all.
Okay. Right. Centrifugation, actually, they use the technique to separate the that DNA part. Where is structure? Which experiment?
So the Hershey and Chase experiment.
This one was for it to make your Hershey Chase. This one.
Yes, sir. This one.
Actually,
The second part of this.
yeah, to separate actually the radioactive matter, right? To separate it, these isotopes, they use a centrifugation method to separate out the DNA.
Okay.
To take actually to extract the DNA from this cell. What they will do? They have used this kind of machine, a centrifugation machine, right? The same centrifugation machine which is used to separate the subunit of ribosomes. Remember?
Okay. Yes, sir.
Clear?
Yes, sir. Thank you.
Okay. Yeah.